Welcome to this explainer. Today we're diving right into the incredible cartography of life by decoding one of the absolute most elegant and powerful visual tools in all of biochemistry. I'm talking about the Ramachandran Plot. Think of it as a topographic map, but instead of charting mountains and valleys, we're charting the literal architecture of life itself.
To really appreciate this map, we have to start with a fundamental paradox. Proteins are massive molecules, right? The covalent backbone of a typical protein contains hundreds of individual bonds. And since free rotation is theoretically possible around a lot of these bonds, a single protein could, in principle, assume a virtually uncountable number of chaotic conformations. Yet, for a protein to actually do its job, whether that's carrying oxygen in your blood or digesting your food, it has to fold into one highly specific, highly stable native shape. So how does it manage to find that one perfect shape amidst near-infinite chaotic possibilities without just tying itself into an impossible knot?
Part 1. The Protein Landscape. Order out of chaos. Let's jump in and see exactly how physical order emerges from chemical chaos. To navigate this crazy landscape and find some stability, proteins rely heavily on hydrophobic interactions. Here's how it works. Pure water forms this highly structured hydrogen-bonded network. When water surrounds a nonpolar hydrophobic part of a protein, it gets trapped in a rigid solvation layer, which decreases the entropy, or disorder, of the water. Now, nature absolutely hates a decrease in entropy. It's kind of like trying to keep a toddler's playroom perfectly organized. It just doesn't want to happen. So as the protein folds, it buries its hydrophobic side chains deep inside its core, away from the water. This releases those trapped water molecules, providing a massive, favorable increase in entropy. And it's this entropic boost that acts as the main thermodynamic engine driving the protein to fold compactly. But that begs the question, what dictates the exact physical shape of that folded core?
That brings us to Part 2, Rules of the Road, the hinges of life. To figure out how a protein moves into that folded state, we've got to look at its strict atomic hinges. Back in the 1930s, pioneers Linus Pauling and Robert Corey discovered something fascinating about the protein backbone. They realized the peptide bond, which is the primary link connecting amino acids together, isn't just a flexible string. Because the electrons between the carbonyl oxygen and the amide nitrogen are partially shared through resonance, the peptide bond actually takes on a partial double bond character. This essentially locks the 6 atoms of the peptide group into a rigid, flat plane. The takeaway? The protein backbone simply cannot rotate freely everywhere. Because that peptide bond is locked and planar, rotation is exclusively permitted at very specific hinges located between these rigid planes. We define these hinges mathematically as dihedral angles, specifically named phi and psi. What's amazing is that the entire three-dimensional conformation of a protein's backbone can be mapped out completely just by knowing these 2 angles for every single amino acid in the chain. If you look at the central alpha carbon, that's the atom right in the middle of our rotating hinges, you can visualize this perfectly. The angle of rotation between the nitrogen atom and this alpha carbon is phi. The rotation between the alpha carbon and the carbonyl carbon is psi. By convention, we say both angles are at 180 degrees when the polypeptide is fully extended. So just imagine these rigid amide planes swinging open and closed around that central carbon, acting like doors on very specific molecular hinges. Part 3. Mapping the Forbidden Zones, the Ramachandran Plot. Because, well, here is the catch. Not every rotation is physically possible. In theory, our hinges phi and psi can rotate a full 360 degrees, from -180 all the way to +180. But in physical reality, atoms actually take up space. They have a specific size based on their van der Waals radii. So if you rotate phi and psi into certain combinations, the atoms of the protein backbone, or its bulky side chains, will physically crash into one another. We call this molecular roadblock steric interference. It's literally the biochemical equivalent of trying to park 2 SUVs in the exact same parking spot. They're going to crash. A protein just can't adopt these clashing conformations.
Enter the Ramachandran plot. Invented in 1963 by the biophysicist G.N. Ramachandran, this graph elegantly maps out all those possible angle combinations. You've got the phi angle on the x-axis and the psi angle on the y-axis. Ramachandran actually calculated the distances between atoms at every single possible combination of these angles. Why? To show us exactly where a protein can and absolutely cannot go.
You can really think of this as a topographic map of molecular survival. Notice how much of this plot is completely white? Nearly 77% of this map represents totally forbidden zones. These are the physical impossibilities where atoms just collide. The blue zones, on the other hand, well, those are the safe territories. The darkest blue regions are fully allowed conformations with zero steric clashes. The lighter blue regions? They're allowed, but only if we permit atoms to squeeze just a tiny bit closer together than they'd normally prefer. Stable protein folding almost entirely happens within these small, restricted blue islands.
Part 4. Landmarks on the map, secondary structures. Now let's see how the structural landmarks we know and love perfectly nestle into these safe blue zones. Take the ubiquitous right-handed alpha helix. When mapped, its coordinates fall precisely into a tightly allowed dark blue region in the lower left quadrant. And this is no accident. In this precise mathematical location, the polypeptide backbone curls into a cylinder that completely avoids steric crashes while perfectly aligning its internal atoms to form highly stable hydrogen bonds every 3.6 residues. It's a masterpiece of biological engineering, and the map predicts it flawlessly.
Similarly, we have the beta conformation. Instead of coiling up, the backbone extends into a zigzag structure, allowing multiple strands to line up and form a beta-pleated sheet. Map the coordinates for these sheets, and boom, they fall perfectly into that highly permitted upper left blue region. Once again, the protein is naturally finding a safe zone that avoids atomic collisions while maximizing structural stability.
We can actually quantify this beautifully. Just look at the exact ideal coordinates for these structural landmarks. A right-handed alpha helix operates right around a phi of -57 and a psi of -47. A parallel beta sheet? It sits at -119 and +113. This goes to show just how incredibly precise and predictable this mathematical cartography truly is. Whenever a new protein structure is discovered, its building blocks invariably cluster right around these exact numbers.
Now, I should mention one really fascinating exception to the map's strict borders, and that's the amino acid glycine. Unlike all the other amino acids, glycine lacks a bulky side chain. Its side chain is literally just a single hydrogen atom. Because it's so incredibly small, it doesn't suffer from those same steric clashes, which gives glycine massive conformational freedom. If we made a Ramachandran plot just for glycine, those blue safe zones would expand dramatically, letting this flexible traveler navigate the otherwise forbidden zones of our map. This is exactly why you'll often find glycine at tight turns and loops in a protein structure. It bends where other amino acids would simply break. On the flip side, an amino acid like proline is locked into a rigid ring, making it the most restrictive of all, basically confined to a tiny sliver of the map.
So why does this matter today? Well, the Ramachandran plot isn't just some elegant piece of biochemical history. It is a vital, everyday tool. As structural genomics pushes to map thousands of unique protein folds using techniques like X-ray crystallography, scientists deposit their findings into the global protein databank. But how do we know if these newly mapped 3D structures are actually correct? We run them through a Ramachandran plot. If a proposed model has amino acids falling into those white forbidden zones, we instantly know there's an error in the data. It's literally the ultimate quality control checkpoint for our understanding of biological machinery. Which leaves us with a final provocative thought. We know that these strict physical constraints dictate the shape of all known life on Earth. But as our computational power grows, and as we continue mapping the massive genomes of entirely new species, how will mastering the rules of this cartography allow us not just to understand nature, but to engineer the uncharted artificial proteins of tomorrow? If we know the exact coordinates of the safe havens, the possibilities for designing custom molecular machines are quite literally bound only by the geometry of the map itself. Thank you for joining me on this explainer. Keep exploring, and I'll catch you on the next one.
Thank you for watching. See you in the next one. Stay tuned.